Hello, my name is Viktor Stolc. I'm a senior scientist at the NASA Ames Research Center. Thank you for the invitation to present our research results on a hypermutation in mice flown on the International Space Station. This presentation is based on a long-standing collaboration with colleagues from NASA, Stanford, SETI, and the Slovak Academy of Sciences. We have been working on the problem of understanding timekeeping in biological systems using genome science technology, high-performance computing, and next-generation sequencing. At NASA, we are concerned with serious health risks that can be attributed to spaceflight, including muscle atrophy, bone loss, liver damage, kidney damage, and spaceflight-associated neuroocular syndrome. As a genome scientist, I naturally think about things that can affect the genome. What about space radiation? We know that radiation can cause somatic mutation, which can lead to cancer, but the International Space Station is protected from such radiation by the magnetosphere. What about other aspects of the space environment, like microgravity, which affects physiology? Are genetic mechanisms involved? It is now recognized that there is a need for fundamental study of somatic mutation in space because of the many varied phenotypes in astronauts and in model organisms flown in space. The results presented here are somewhat surprising and have even implications for the topic of sleep in space, which I hope you may find interesting. One of the most important take-home messages from this presentation is that time is an essential non-random variable of the information content and processing functions in cellular systems. Please keep this in mind for the rest of this talk. I'm sure that everyone in the audience will appreciate that sleep is critical for human health and is required for all mammals. The timing and duration of sleep are controlled by circadian system, which keeps a 24-hour internal rhythm that synchronizes to environmental stimuli. All organisms from mammals to prokaryotes have an internal clock adapted to the Earth's rotational schedule of 24 hours. By contrast, the International Space Station orbits within 93 minutes, creating a 93-minute day-night cycle with different environmental stimuli. Therefore, it is natural to ask, what is the origin of biological timekeeping on Earth, and is it different in orbit on the ISS? How is biological timekeeping altered in space in humans and organisms that are used to study circadian systems on Earth? Another way to put it is, what time is it for sleep in space? So it is becoming increasingly clear that there is a link between metabolism and circadian timekeeping. If that is the case, it raises the following question. Might some of the products of metabolism serve to reinforce or influence circadian timekeeping? In essence, it is a chicken or the egg dilemma. Reactive oxygen species serve a timekeeping function as switches of various cellular processes and physiological rhythms. The best candidates for timekeeping functions are reactive oxygen species oscillations, such as hydroxyl radicals, which are produced as byproducts of bioenergetic metabolism. A complex network of coupled multiple feedback oscillators, such as temporally controlled RNA transcription of periodic proteins and enzyme activities, are coordinated by the redox cycle. These result in various biological phenotypes, such as proliferation, differentiation, autophagy, immune functions, and aging. To counteract excess ROS, cells contain antioxidant systems, including peroxyredoxin enzymes, which protects against oxidative stress. These peroxyredoxin enzymes, in essence, serve as markers of circadian rhythm systems. Mutants with defective peroxyredoxin enzymes exhibit increased somatic mutations caused by ROS. More about hypermutation as a consequence of gene activity during the oxidative phase of the redox cycle in the next slide. Studies in yeast provide a wonderful example of how the redox system is cyclical. Even though yeast don't have lungs, they consume oxygen in a cyclical manner, as shown on the top left panel, much like breathing. Oxygen consumption oscillates between low and high respiration, which corresponds to oxidative and reductive phases of the redox cycle, respectively. The redox cycle generates an oscillating pattern of gene expression as shown in the bottom left panel. Another way to say this is that some genes are expressed in the oxidative phase and different genes are expressed in the reductive phase. The graph on the right shows that genes transcribed in the oxidative phase accumulate mutations significantly more than genes transcribed in the reductive phase. The mutation frequency is plotted on the y-axis. Therefore, environmental stress that induce ROS, such as spaceflight, have the potential to mutate DNA and disrupt biorhythms. One of the most interesting points about this data is that ROS induced by the environment enable a coupling between heredity and the environment. Therefore, studying life processes in space provides a window of opportunity to unravel the timekeeping functions at a fundamental level. Based on the timekeeping functions of ROS oscillations in yeast, we explored the biochemical effects of spaceflight aboard the International Space Station on the redox cycle in mice to determine if environmental stress affected their genome sequence and circadian rhythm. A special habitat is used on the ISS called the Rodent Research Hardware System Platform, which was designed at NASA Ames Research Center as shown on the right panel. We examined a relationship between metabolic activity, sleep, and metabolic rate in mice flown on the International Space Station for only about 40 days. Here is a movie of mice activity on the International Space Station. This is an unusual environment for the mice, but they still exhibit some range of normal behavior such as self-grooming, locomotion by using their momentum, and grabbing onto the cage to move around. The environment that they are in makes it very difficult for the animals to sleep, even though they appear to be well adapted to microgravity. This becomes apparent after about 14 days on board the ISS, as mice become sleepless and hyperactive. Just to clarify, the music about time was added to the movie, and mice did not lose sleep over it. We can use a simple graph, though, to quantify the activity of the mice, and as you can see from the blue line, they sleep very little on the ISS. Most importantly, there is a dramatic increase in the activity of mice in space on the ISS in comparison to ground control mice, represented by the red line on the graph. Quantitative behavior activity assay revealed that mice became sleepless in about 14 days on the ISS. These experiments that were done in the animal habitat on the ISS are insufficient to address the question of circadian rhythm disruption due to elevated ROS. There may be additional confounding variables in spaceflight. To analyze another important consequence of reactive oxygen species elevation, we examined the rate of somatic mutation in mice flown on the ISS as shown in the next few slides. So for the experiment, 16-week-old mice were sent to the ISS with equal number of mice kept in nearly identical rodent habitats on the ground at the Kennedy Space Center. To determine the rate of somatic mutation in spaceflight, RNA sequences from different tissues of mice were determined by next-generation sequencing. The web links for the NASA spaceflight samples and data collections for these experiments are shown at the bottom. Somatic mutations were determined by a novel analytical pipeline developed at the NASA Ames Research Center. Generally speaking, somatic mutations are rare events, and finding them in the RNA sequence data is like looking for a needle in a haystack. To accelerate identification of the spaceflight-induced low-frequency somatic mutations in mice, also called de novo mutations or single nucleotide variants, SNVs, a novel software was developed for high-performance computing at the Ames Research Center. The somatic mutation analysis pipeline processed a single mouse genome for single nucleotide variations in about an hour. The somatic mutations induced in mice on the ISS relative to ground controls from over 500 tissue samples were mapped from the RNA sequence data using approximately 100 terabytes of data in about 1 week. Using next-generation sequencing, the entire genome can be sequenced quickly and efficiently to study somatic mutations. Over the last 15 years, next-generation sequencing enabled studies of somatic mutations have shed light on the development of precancerous conditions, cancer, inflammatory processes, and degenerative diseases, as well as sleep disorders, which are all relevant to deep space travel. To compare the performance of the NASA Ames somatic mutation pipeline, or the figure of merit, it was compared to the highest performing pipeline in the FDA Truth Challenge in precision and specificity. Our pipeline achieved 99% precision and 99% specificity, which is equal to the best available variant calling pipeline. So we're very proud of it. If nothing unusual were going on, we would have expected 1 or 2 somatic mutations during the period of the experiment, which lasted 40 days on the ISS. In fact, we observed many thousands of mutations in each tissue that was examined. By comparison, essentially no somatic mutations were detected in the ground control mice. As shown in the Venn diagram, some somatic mutations were unique to the specific tissues, some were shared, and several of the mutations identified were common to all 4 types of tissues that were analyzed. These are the hotspots of variation in the mouse genome. The International Space Station induced mutations led to low level of genetic mosaicism in different tissues, and some of the non-synonymous mutations likely result in loss of functions in areas of physiology relevant to spaceflight risks. In comparison to the known mutations that accumulate over the lifetime of an organism, the International Space Station induced mutation rate is 5 orders of magnitude higher than on Earth in every tissue examined in just 40 days. Additionally, the genes that are expressed the most exhibited the highest rate of somatic mutation. This raises the question, are the somatic mutations completely random? Well, the answer is no. It turns out that guanines are substituted more frequently, and the data are shown on the next slide. We know from chemistry that guanine is the most easily oxidized of the nucleotide bases, a process that can occur because of endogenous production of reactive oxygen species. Out of the 12 possible base substitutions, the preference for guanine substitution was observed in all but liver tissue types, which is consistent with the notion of the elevated reactive oxygen species generated on the International Space Station. In summary, a high rate of somatic mutation was observed in mice that spent 5 to 6 weeks in space aboard the International Space Station. Non-synonymous mutations, or mutations that affect the amino acid sequence of the proteins, amounted to approximately 1/4 to 1/3 of all mutations observed. These are the mutations that count because they can change the protein functions and phenotype. Space radiation exposure by itself, however, cannot account for the increased rate of somatic mutation that were observed in mice on the ISS. The radiation levels on the ISS are simply too low. Many aspects of spaceflight environment may have contributed to the observed increase in somatic mutation rate, including weightlessness, stress, and other environmental factors. Elevated carbon dioxide aboard the ISS was possibly a major driver of the increased somatic mutation rate that was observed. Elevated CO2 can result in generation of reactive oxygen species, which can lead to somatic mutation, especially involving guanine base changes. This hypothesis is consistent with the observation that CO2 increases somatic mutation rate in ground-based cell culture studies. Additionally, sleeplessness in mice on the ISS can also be due to neurons responsible for arousal that are directly triggered by the elevated CO2 and cause mice to wake up and maintain vigilance on the ISS relative to ground controls. In humans, the retention of CO2 in the blood is linked with sleep disorders that affect breathing, especially sleep apnea, central hyperventilation syndrome, and obesity hyperventilation syndrome. The take-home messages from this presentation are that stress of spaceflight and likely other environmental stresses affect the timing of the redox cycle. Oxidative stress results in loss of function somatic mutations, and circadian rhythm is perturbed by oxidative stress. Based on these results, NASA needs to consider the following next steps. The rate of somatic mutation in astronauts in space needs to be studied. So far, we only have data for mice. I believe that it is time for NASA and other space agencies, including the private enterprise going to space, to consider metabolic control methods for the purpose of achieving a hypometabolic state. A hypometabolic state will likely result in a significant reduction of somatic mutation rates and may help to prevent some of the deleterious effects of space travel. With this, I would like to thank you for your interest in this presentation, and please feel free to reach out to me if you have any questions.